I was here 20 years ago. So thank you very much for inviting me. I really enjoy being here again. And we'll do this together with Don Klinkenberg, who did a long time ago a PhD with me. And he will introduce himself further. So what we want to talk about is really what the last talk almost ended with, is the relationship between more empirical people and modelers, and how we can work together, and the way that we really need to use the knowledge of both to get to better models. If this works. Oh, then I have to put this in, of course, otherwise it's not going to. Yeah. See where, yeah. Okay. So what I wanted to start with is something that Hans Metz once said it is noted in a little booklet. And real life is but a special case and not necessarily the most interesting one. And what he really means is real life is just a special case of all possible models. And sometimes listening to modelers, that's the feeling that you get. And as somebody who comes from a more empirical background and very much interested in modeling, I sometimes have the feeling that even with these complex models, the reality is not in there. I mean, reality is just another model that still needs to be designed. So that's something that will come back in this. So what we really want to do is to find out where the reality fits with the models. And the reason that we want to do that is that we want to control, eradicate, or mitigate risks from infectious diseases. We want to, in some way, manage infectious diseases for the good of people, eventually, and animals maybe intermediately, plants, and so that's what we are talking about. So that requires models with a kind of optimal balance between mechanisms, biology, and tractability. And so that asks for a cooperation between mathematicians, modelers, and more experimental people. And we're talking about veterinary epidemiology, so we'll get some examples where this cooperation really failed, and we want to discuss about that a little bit. So you want to have maybe a correct description of the observations. So this could be a hat, but of course you will also know what causes these observations. So in epidemics, the issue was and is that really we cannot observe the contacts that cause the transmission. I mean, it has been mentioned today already. Maybe for infectious diseases, you can say, okay, this kind of this kind of sexual encounter between two people is the cause of the transmission. But often we don't know about the real contact, and so that really remains a problem. And we have been trying to do experimental work on animal diseases to find out more about that, and I will briefly come back what I told 20 years ago, and at the end I will go one step further at what we are thinking now about how you can model contacts and how you need to improve that. So one way that contacts are often modeled in these models is proportional to density, and then defining this density, I mean that's kind of proxy, a kind of measure for the number of contacts that you would have. So you say, okay, this density, and then if you have twice the density, you have twice the number of contacts. Then defining and measuring this density becomes the real issue for extrapolation. So in the 93 meeting, I tried to explain that for animal husbandry, we can often assume constant density. Animals are kept basically in very much the same way. So if you have 200 dairy cattle or you have 400 dairy cattle, everything is just multiplied by 2. If you have twice the size of the areas, twice the size, the number of milking equipment, the number of feeding stations. So you're basically the same density. So 2 individuals in the population, the number of encounters of an individual in a population will roughly be the same. And so we said, okay, extrapolation of transmission in vaccination experiments, that's what we were doing then. We were looking at the effect of vaccination on the transmission of an infectious disease. And we were wondering, how can we extrapolate what we have found in a group of 10 to a group of hundreds of pigs? And so one extreme would be that to say, okay, the R0 goes up with a factor 10 because you have 100 times the number of pigs. Or you could say, okay, basically they have all the same density, and if you model in terms of numbers of pigs, you can write Si divided by N, and so you end up with R0, which is not dependent on the number. So the same R applies. And I showed then this experiment. So this was an experiment with 40 pigs. So basically we showed in 10 pigs that the R0 was 0.5. And then we did an experiment with 40 pigs. 20 pigs were infected and 20 contact pigs. And this is the distribution of contact pigs under the different assumptions. And this was the observed number of contacts. So we concluded that indeed you could assume that R0 does not depend on the population size. So the goal was eradication of pseudorabies virus, also called Oyeski virus. It's a herpes virus of pigs. And the eradication was officially announced completed in January 2009 with this vaccine. So we were able to model this vaccine and show how the prevalence would go down, and we would also able to say beforehand, okay, this kind of things still could happen, small outbreaks in certain farms, and it went down a little bit faster than we predicted, so that was lucky, and so it was finished on January 25. So we want to discuss very briefly a couple of examples. The first example, and you all will know why I want to discuss this example, talking about modeling and about people working in the field, veterinarians, was the foot and mouth disease outbreak in 2001 in the UK. So there was predictive modeling to inform policy, and one of the conclusions was that the current policy is not going to work at some stage, and preemptive culling of farms is necessary to stop the further transmission. And then a big controversy between modelers and veterinarians arose about this, where I just cite a couple of papers. If you go to look in the literature, there's quite a lot about this. In fact, I was here in London at a meeting of DEFRA afterwards, where modelers and veterinarians came together. And I know both John Wildsmith and some of the modelers quite well, so that was kind of awkward situation. But I mean, there was a lot of discussion about it. So Kitching et al. Published a paper, and he said, yeah, there were really unvalidated predictive models. And Ronald Cow, of course, modeler of background. All approaches are useful tools, but have limitations. And Morris, New Zealand, only simulation model can accurately represent the fine scale heterogeneity. And so one of the basic discussions here was that this preemptive culling was just blanket culling of all the farms and all the animals that were susceptible for foot and mouth disease. And veterinarians thought, okay, we can much better in the field say these farms should be culled and these farms should not be culled, and it's not necessary to cull all these farms. And that was one of the big controversies that arose. And so the question was, was the preemptive culling necessary and was it sufficient? And so maybe it was not always necessary. I mean, you can in hindsight say that certainly some of the farms probably had not that kind of high probability of becoming infected. And one of the, I mean, there are many kind of heterogeneities, and there was lack of knowledge about these heterogeneities and how that would work out in terms of the transmission. So that was the point. And so I will take one example of the heterogeneity, so that's the species differences. Just as an example, and also because we have been doing experimental work, so our approach is more to look at controlled experiments to find out about these kind of things. And because of the statistical nature of the spatial relationship, it's probably certainly, yeah, that's a nice one. That's it. That's when you stand here, you see it. It's probably sufficient. I mean, it's almost certainly sufficient because it just says, okay, you infect so many other farms, and if you cull a certain range, then most farms that get infected are in the neighborhood, and if you cull all the farms in a certain neighborhood, then you will, on average, infect less than one farm, and so you will end up with stopping the epidemic. And here I take just one example, the species differences. And so I would give some estimates of the species transmissions parameter. And so in foot and mouth disease, there are basically three important species: sheep, pigs, and cattle. And so you basically have, in terms of parameters, if you have a transmission rate parameter, you have a 3x3 matrix. And so I try to reconstruct that 3x3 matrix. We know quite a lot about the sheep-to-sheep, pig-to-pig, and cattle-to-cattle transmission, but less about the interspecific transmission things. So pigs to cattle, we don't have any reliable estimate, but from the work that's done here in Pirbright, this is very high, this transmission. Pigs to sheep, we have this estimate based on data from Pirbright. Sheep to sheep, on the same data of Pirbright, we estimated 0.15 per day. Another publication that we did, Karen also did a PhD with me, she estimated 0.105, so we took 0.1 per day. I mean, you just want to get some feeling for the order of magnitude, so I don't want to put their figures with many decimals behind. So pigs to pigs is about this, and cattle to cattle is about this. I don't put anything about the vaccination, but basically cattle is well protected by vaccination. Sheep and pigs are not very well protected by vaccination, but that's another part of it. So if you look at the between species transmission parameter matrix, 3x3, then you can try to split it in infectivity and susceptibility. And if you then assume separable mixing, then you really have 5 different parameters in this matrix to fill this matrix. So you take one. I will show that in a minute. And so we need one more.
Sorry, what are these numbers?
These are the betas for the transmission from cattle to cattle, from sheep to sheep, between animals, and the 50.
But cattle to pig, does it mean you put one cow in a pig farm to let it run around, or what does it mean?
Yeah. No, not necessarily, but the estimate is if you would have one cow in a population totally existing of pigs, it would in fact, on average, totally consisting of susceptible pigs, it would on average infect per day 50 other. It's not here now, it just says hi, but
1,000, if I remember right from your
yeah. So, but you see here, pigs are very infectious. This number is quite 50. They infect 50. So in a few hours,
And the other 4 places in this matrix are very small.
No, the other places I'm still going to fill. So if you can write it as separable, you can write it as a product of some susceptibility and infectivity. Then you need 5 parameters to fill the whole matrix. So if I have 5 measurements, then I can fill it. And I only have 4 because I don't trust this one. And so what we did, we made another one. So this is the experiment, 2 sheep and 1, so that's really the experiment. 2 sheep and 1 cow, and then many replications. So that's a PhD student of mine, Carla Bravo de Rueda, who did this. And the sheep were inoculated and the calves were contact. 4 out of 10 calves got infected. You can do the whole exact final size thing, and then you get this as the maximum likelihood estimate. You can just fully do the, so this is the maximum likelihood estimator for this experiment for R. And you can fill it in. I have it here, that the R is something like this. So they're about infectious for 10 days, so not all the signs. So it's again about 0.1 per day. So now we have this matrix, and now we can fill in all the susceptibility and infectivity. I will do that very quickly. So we assume separable mixing. We start with 1 there. That's very clear. So clearly sheep and cattle are as susceptible. I mean, that's what you could have guessed, of course, from this. I mean, that's the obvious thing. And so then we have this one, we can calculate that. So pigs are 25 times as infectious than sheep, than cattle, many more times more infectious than sheep. And so we get this. So basically now we can fill also the other elements. So this is the how this matrix looks like. And so what you see is that pigs are very infectious, but very low susceptibility. I mean, this transmission and that transmission is about the same order of magnitude, but it's of a different composition. Pigs are much more infectious, but also much less susceptible than cattle. And sheep are even much less infectious. That depends on your assumption of separability. Yeah, very good. So that's something to. So then, if you look at the basic reproduction ratio, you can make it into a next generation matrix. Because it's separable, the R0 is just the sum of the diagonal. It's the trace of this matrix. Because it's separable, this matrix, it's really one-dimensional. So only one of the eigenvalues is non-zero, so then the sum of the eigenvalues is the trace, so the eigenvalue is the trace here. So this is just an example. So you have to realize that this is, of course, all between animals. And eventually, of course, you're going to look between farms. And there are some steps to be made, but it's helpful in the modeling to think about the differences between the different animal species. So separable mixing is really a good question. We should, again, we should always look at this. At this moment we don't have contradicting data, but we really should look at this maybe more. Transmission of foot and mouth disease is mainly between cattle to cattle, especially when you look between farms. And pigs are much more infectious, but they are very difficult to infect. So if there are no infected pig herds, the chance that pig herds get infected is very, very small. So you don't need to do anything about the pig herds as long as the infectious herd is a cattle herd. If the infectious herd would be a pig herd, then you would have a problem. Then you would need to do something about the pig herd. But if it's a cattle herd, you don't need to do. And so we did also experiments to show the effect of distance, and I'll come back to that later. But I mean, because of time, I won't tell much about that. If pigs get infected, they infect more sheep and cattle than cattle and sheep themselves would infect. I mean, that's clear. And sheep are as susceptible as cattle, but much less infectious. So that leads to also for vaccination strategy to vaccinating cattle and cull pig herds when they get infected or have a dangerous contact. And sheep have to be monitored to avoid moving infected sheep. Oh yeah, this is that I wouldn't forget to say that. I did say that already. So with the issues, of course, that this is between animals, but we really want to look between farms. So this is your slide.
Okay, it's working. My name is Klinkenberg. I'm working at the vet faculty in Utrecht with Hans and other people, of course, on modeling and statistics and on experimental data of animal diseases mainly. What I'm going to do here is give 3 examples of infections that have played a role in the last 20 years. I did looking in the literature and focusing on what Mart was already said in the introduction about these contexts that we do not observe. And it's and the first example is the BSE. And in the BSE epidemic, the main problem was that there had been, at the time that the epidemic started, or at least that the epidemic was detected, there had been already many unobserved contacts in the past, and the big question at the time was how big is the problem going to be. So in 1986 it was first identified, and in the first year, so slowly more and more cases came, and in May 87 epidemiologists at the CVL, the Central Veterinary Laboratories, was called for help. John Wildsmith, and in about half a year he established the fact that the epidemic was probably due to feed, and most likely meat and bone meal that was rendered, so it was offal of cows and sheep being fed to the cows, and that was the most likely cause of the epidemic. And back and some calculations based on incubation times led him to estimate that the exposure must have started around 81, 82. Then a couple of months later, it was described as an extended common source epidemic. The hypothesis was at the time that it was caused by the scrapie being fed to the sheep, and that a change in the rendering method around 81, 82, in the way these cattle and the sheep were dealt with before being fed to the cattle again, that the change in that method increased the infectivity, so that must have caused the epidemic. So this extended common source epidemic, at the time there were about 60, maybe 100 cases per month, and because the idea was that it was caused by scrapie, I mean, already in a couple of months, maybe 1 or 2 years, that it was found out that it was actually also the cattle being fed to the cattle which caused a real big multiplication, but at the time that was not really known yet. So the idea was that number of cases about 60 per month. We've had them for about 6 to 7 years. So okay, we have to stop feeding this meat and bone meal, but there was not a real very high urgency. So in a couple of months it was implemented, and there was no large-scale callback of feed or something to show the urgency of the problem. And in that way, in this estimation of the number of cases that were to be predicted, and the fact, and it was thought that this would really stop the epidemic, What was underestimated was, well, first the fact that it was the cattle meat and bone meal being fed to the cattle again, and so multiplying and multiplying the infectivity. So in fact this 60 turned out to be 3,000 later on. That there were still stocks present with the producers on the farms, and that there was cross-contamination with the feed producers and cross-contamination on farms. And that was because this meat and bone meal was still allowed in pig feed. So that caused this cross-contamination. Well, a couple of years waiting, but then in March 1991, and that's what I want to focus this BZ topic on, is the fact that the first case born after the ban was detected, and it was expected at the time that these cases would not develop, because stopping this feed ban, installing this feed ban should have stopped new cases. So you see, well, you don't see it on the scale, but this is the development of all the cases, and at this time the first born after the ban case developed, and also increased in time. So whereas it was originally thought that it was under control, it appeared not to be under control. At least that was the fear at the time by the people, not knowing that these BABs only indicate that the basic reproduction ratio, so this backfeeding, is above 0, whereas of course we need to know whether it is above 1 to know if it's under control. But in the communication from the people doing these very early predictions to the public and to the policymakers, this was not explained very well, and it was suggested that this meat and bone meal ban would be 100% effective. So now more and more BABs developed. It appeared not to be 100% effective. So then it was feared, is the epidemic under control at all? Well, then modelers stepped into place, and in 1996 a publication by Roy Anderson and some other people in this room, I think, appeared in Nature, where they did the first real proper back calculation model, where they explained the incidence, so it's the case incidence, number of diseased BSE cases developing at HU born at some T0. They expressed this incidence in terms of all kinds of relevant quantities and distributions, like the survival, maternal infection probability, the age at infection, the feed risk, so that's takes into account the implementation of this meat and bone meal ban, incubation period distribution and reporting probability. And based on these calculations, these models, by the way, were inspired by all the HIV back calculation modeling that was being done at the time. So in that model described what had actually been going on. So this was the yearly incidence that had been observed at the time. In 1995 we are here, so the top is 35,000 BSE cases, and here you see the same blue curve here, but then on top of that the actual number of infections that had been developed and the number of new infections that had still developed after installment of the meat and bone meal ban here. So based on this, well, 1 year later, interestingly, in this paper by Anderson, where they did this prediction or this analysis, R0, or the basic reproduction number, was not mentioned at all. But 1 year later, Neil Ferguson confirmed the analysis by doing lots and lots of sensitivity analysis, and he gave R0 estimates indicating that horizontal as well as vertical transmission was very, very low, so that the epidemic would come to an end. This is the prediction that was done by Anderson in that paper. So the blue line is the prediction with the prediction interval, and the gray line is the actual case development that turned out to be that I got from the OIE site. So it was pretty accurate, I think. So the conclusion on the BSE is that this bad panic was caused by false expectations, so by not perfect communication. And I think what we should learn from this is that we should communicate the expected effect of control, so that if control is effective, it doesn't mean that the reproduction ratio is equal to 0, but just that it's below 1. Of course, the smaller the better. And that new cases, and that's an important message, that if new cases develop, even after implementing the control measures, that does not mean that the epidemic is not under control, and that even new cases are expected. The second example that I would like to discuss is blue tongue, and in this case the unobserved contacts are the contacts between the sheep and cattle as a host and the vectors. So blue tongue is a vector-borne disease. It is transmitted by midges, and but not by all midges, by only some species, some Culicoides species. And basically the home range of these vectors determines where the contacts occur, so where there is a risk of blue tongue emergence. And that's basically not too much to the north and not too much to the south. So none of the known vectors were present in Europe. But still, in 2006, there was a huge blue tongue epidemic in the north of Europe. It started in the Netherlands, it spread to Belgium, North France, and Britain. So there are lots of cases. In all the colors represent months in 2006. Then it was thought, well, maybe it's over after the new winter when all the vectors carrying disease die, but in the next couple of years there were still epidemics. And it turned out that a different Culicoides species, Culicoides obsoletus, appeared to be a capable vector as well. It was not known at the time. So then the question arose, well, if it can occur here, where else can we expect blue tongue outbreaks? So where and when are outbreaks in principle possible? So that is determined by the presence of the vector and by sufficient climatic conditions, because all kinds of characteristics of these vectors, like biting rates and survival, depend on temperature. So it should be that the vectors are not only present, but that they should survive the incubation period, and that they should bite sufficiently frequently to transmit the infection. And that started a series of studies where risk maps were being made and predictions were being made to predict where outbreak of blue tongue could occur. And basically there are two approaches. You can simply use statistical models, not doing any modeling, or you can use mathematical modeling using R0. And we'll give examples of a couple. So first, statistical modeling, you can just answer, well, if there is blue tongue, you can do this in an endemic situation. How does it relate to all kinds of climatic and landscape factors? So this is an example in Corsica, where you have basically it's a case control study, and it was found that blue tongue presence on farms depended on all kinds of things like sunshine, farm type, landscape factors, and then you can make a prediction of where on this island you could expect other blue tongue outbreaks. Another way of doing it is looking within farms and trying to relate the observed incidence to all kinds of predictors, and then predict incidence based on rainfall and temperature, and see that your prediction pretty well reflects what actually happens. So that was modeling blue tongue outbreaks in relation to climate. You can also model blue tongue directly in relation to presence of the vector. And that can also be done in a case control kind of setting or within farms. So in a case control kind of setting, it was quite interesting in the US, in North and South Dakota and Nebraska. It turned out that in the end, presence or absence of the vector was not a good predictor of blue tongue outbreaks, but latitude and altitude were. So probably, so I think it indicates that there's maybe something wrong or imprecise in measuring how many vectors there are, whereas maybe this latitude and altitude also predicts, and much better predicts, whether there are vectors and which can transmit the disease. Also here you can look in time series, so it's a couple of years following a single herd, where the increase in numbers of vectors always precedes the increase in incidence. So a different way, that was statistical. You can also do it by mathematical modeling, so using a formulation for R0. This is a formulation for R0 used for many vector-borne diseases. And many of these parameters are actually individually measurable, such as vector mortality, incubation periods, transmission probabilities, and biting rates. But the major unknown in this expression is the vector-to-host ratio. So basically the major unknown is the number of vectors. And the problem, so to get an idea of how the number of vectors is distributed over space, you have to get back to statistics. And in fact, you're doing more or less the same thing, only there's an R0 step in between. So we model vector abundance, also trapped vectors in relation to climate and landscape. So you can do it without R0, so this is distribution of two types of vectors in Spain, and indicating where most of the vectors are, which would also indicate where the highest risk of blue tongue is. And here it's a study in which for all the different trapping sites, the number of trapped mosquitoes was translated into an R0, and in that way making a risk map for blue tongue transmission. But the problem is, I think, in this second approach, so using modeling, is that the validity of these trapping data. So the major unknown in this R0 formula is the vector-to-host ratio, so the number of vectors. And there are loads of studies indicating that there are problems with measuring these vector quantities by trapping them. For instance, it depends on where you put the traps. You put them close to farms or a little bit further from farms, and then the vector densities really change. And even then, if you go a little bit even further from the farm, but until to the edge of a forest, then the number of vectors increases again. So there's lots of spatial heterogeneity on a slightly larger scale, but also in a very small scale, because also within the farms there's already very high clustering. There are really these clouds of midges, probably, which are just not very well estimated by using these traps. And then the question is which kind of traps to use. So different traps give different trap counts, and if you really look at the amount of vectors captured in a trap and looking at how many bites individual animals receive, which is also done, then you see that these trapping underestimates the amount of bites that the animals get, especially if it's very windy. So I think that simple associations in this case may work better than a complicated model. So directly linking from climate to where can blue tongue occur, maybe even be maybe more useful than using the intermediate modeling step. So what's the additive value of? So in this example, I question the additive value of mathematical models, but I think it's not a unique example. Because I think in many cases we use statistical models instead of very mechanistic models, because they simply work better, especially when you want to do kind of short-term predictions. So then you are more interested in describing the correct outcome than in really describing what is the underlying mechanism. So we want to know. So if the description of the head is sufficient, for some applications it is, then it's not necessary to know that it's actually an elephant being eaten by a snake. So that's these risk maps, but in fact transmission kernels is also an example of a statistical description of something where we can do short-term predictions but not really understanding what's being done. And transmission kernels are the final example that I want to part of the final example that I want to discuss. That's avian influenza, and there I want to discuss unobserved contacts between farms. Because in avian influenza, so generally it's thought to be that the pig is more or less the mixing vessel for avian influenza strains, for pandemic strains. But when you really look at avian influenza, all the virus strains circulating in wildlife, and I think it's poultry that's really the bridge from that takes all these strains from wildlife and permits contact with other animals, well, especially with humans. So that's why the control in the poultry is very important. Within the farm, avian influenza is generally controlled by culling animals. So the key to control is how to control the contact between farms. And for that we need to know how it is transmitted. So which are the unobserved contacts that result in transmission. So I'm going to discuss a couple of approaches that give some idea of it, and then slowly move into the future, going back also, the first approach, case control studies. Very classic approach, looking in endemic or situations or after epidemics, just comparing cases and controls, and then you find associations with, for instance, farm types. So the odds ratio for layer farms is twice as high as for broiler farms to be infected. So you do not get a per contact risk, and it only gives an idea of what the control could do, but it's not very precise. An exception is one study being done very recently based on H7N7 epidemic data in the Netherlands, where the case control was actually done on not on farm by farm base, but on contact by contact base. I will come back to that study later. So the second approach is using a transmission kernel, describing the transmission probability as a function of distance to the farm, and then you can indicate where the high-risk regions are. That's done for a couple of infectious diseases with farms, but this is a very descriptive. It's not mechanistic. Or dynamic. It's only a statistical approximation. So in fact, you are looking at the previous epidemic, and you cannot look at effective control. The third approach is looking at contacts of individual farms, for instance, through questionnaire data. But then you only know how many contacts farms are making, and you do not know how these contacts are related to infection. Fourth approach, which is really nice but has the same problem as the previous, is that you look at all the registered contacts in a network and see how these networks are, and then you can investigate this structure and look at the role of catching companies that catch the poultry before they go to slaughter, the role of company affiliation, of slaughterhouses that are shared by farms, and you can make networks of all the farms that are linked by slaughterhouses or by other types of contacts. And even more nicer is that if you have the data in a dynamic way, so if you have connectivity data in a series of years, that you can simulate these contacts and simulate epidemics and also look at the role of these different players. But the problem, as with the previous example, is that you do not know the transmission probabilities per contact. And that's exactly what we need if we want to look at the effectiveness of control. So then, going to the future, there's probably a role of genetic data, and also already in the previous set of presentations this was discussed. So we could actually reconstruct exactly how outbreaks have developed if we have data of all the farms that were part of such an epidemic. So in the H7N7 epidemic in the Netherlands, so the colors just indicate spatial clusters, and when you only use the genetic data for the hemagglutinin gene, then you can nicely see that there were two long-range contacts from this yellow cluster where it all started to the red part. And by this reconstruction of the epidemic, you can really look at you can identify individual contacts, individual transmission experiments. And then in the step next to it, you can, if you have this case control study per contact, so you can look at what the probability of transmission for the individual contacts is. For instance, you estimate that it's 30% for egg transports, then you know how many egg transports there were, so there are 2 cases caused by egg transports, and then you can validate this by looking at these reconstructed transmission trees from the phylogenetic data and really indicate which contacts that must have been. That's very nice, and I think that's a nice development. Finally, you can also reconstruct the transmission tree using spatial, temporal, and genetic data all at once. Then you get, so these are all the arrows linking all the different farms, and then you can look at the wind direction at the most likely moments that these transmissions had taken place, and see what proportion of the of transmission is most likely to have taken place through wind. And if you do that, then they come with an estimate of two separate studies, about 20% of the contacts must have been due to wind transmission. And then let's take over again.
Okay. Sorry. So, everybody was awake again. Ending up our presentation, I want to say something about modeling is it working? Yeah. So modeling the contacts based on experiments. So transmission models, almost all the models that you see, take implicitly make the implicit assumption that the transmission is instantaneously, so that the chance of you getting infected depends on the number of infected individuals present at the same time. And so that's something that you really can question, because in fact most of these infections go through an environmental stage. So you sneeze on this table and then somebody touches this table and it gets infected. And so the whole question is, so what exactly happens in this environmental phase? And so one way to think is that it could be instantaneously when the environmental stage is very short, if there's a kind of transmission through the air, and then the stage in the environment is very short, so you can ignore that. But that's not necessarily the case. So we want to look at that more in an experimental setting. And so we have been thinking about how can we tease these things apart? How can we understand this? And there, if you are at the same place at the same time, of course you can infect another individual. But there are some situations where you're not at the same place or not at the same time, and you still infect other individuals. So, see if I can do this. No, I cannot do this from here. I have to do it from here. Just okay, now I've lost it. Let's see where. Oh yeah, oh yeah. Yeah. So I want to have this one. So this is really the assumption that you infect somebody through the air. And so what you have is that you have a certain range around you, but it's still instantaneously. So that's one way of looking at it. In fact, what is really happening is that what's really happening is something like this, so that you leave kind of a trail of infectious material behind. So you cannot only infect somebody who is at the same time at the same place, but you can also infect somebody who comes after you. I can sneeze on this table, leave the room, and somebody can get infected here. And so what we were thinking is, can we experimentally look at this in more detail? And so there are really two kind of situations that you can do experimentally. So you can have infectious animals in a cage, take them out, and put other animals in and look whether they get infected. Or you can have infectious animals in one cage and a recipient in a distant cage. And I want to talk about this latter situation. So you have an infectious animal in one cage, and you look at the cage at some distance. And so what is really happening there? So what will be happening? So the infectious material has to go from one cage to another. And clearly, if this is going through the air, so we take an example of a certain bacterium in poultry in chickens, and the distance between the cages is 75 centimeters. So if it's airborne, then it's almost going to be instantaneously. It's going to be at a constant rate, maybe a slow rate, but it's going to be, the transmission is going to be instantaneously, because airborne is something like half a meter per second, so 75 centimeters is a question of a few seconds. And so what we expected is that, I mean, it's airborne, but it has a very low probability. So that's attempted all the time. So this Campylobacter, you have to know it's excreted by these chickens all the time after they have become infected in very high quantities. So what you expect is that there's a low probability all the time that the animals get infected. And so the number of cases is proportional to the number of infections at this moment, but it's a very low transmission rate. But this is how the data looked like. So this is something that Bram van Bunnik did in Lelystad. So this is the number of days, and you see here 1, and then there 1. So it takes quite a long time before these animals get infected. So if it would be some kind of Poisson process with a very low rate, then you would expect some kind of linear cumulative line here, starting in 0, 0, and then being just one line. And so that is clearly not the case. It starts much later, and so we try to understand why this happened. So in discussing this, we counted a paper where Don was involved with, and you here see the same kind of problem. So these are animals from another bacteria in pigs in this case, with a meter distance between the stables. But you see here also that a few days, and clearly it's not a linear relationship through the origin. It's some other, there is some delay involved. And so the thing is, how can we explain this? So it's not because the animals are not infectious immediately. If you have put animals in the same cage, they get infected immediately. So, and so what we were thinking is that survival of Campylobacter is very low. So if you would have a kind of random displacement of infectious material through space, like a random walk of infectious particles, then arriving at a certain distance is much later when you have a high mortality on the route than when you have low mortality on the route. And so one of the things that we have been thinking is, could we compare it to another bacterium which has much lower mortality? So this is the kind of model. So you have a kind of, so the random walk over space then translates into a kind of diffusion, kind of Gaussian distribution over space if you would start from one point. And so, so in this case, it's kind of different than the normal diffusion models. So because here there is this minus sign. If there is this plus sign, then you get something, if you have an R0 of above 1, if I'm still allowed to say that, then you get some kind of traveling wave at some speed from. But now what you have is that you have just a source term, and this is negative. And so then you have a solution. Then there is a certain distribution. That's how far it will get. If this alpha is larger than 0, if there is some mortality, then there is a limit to how far you can get. You have a source cage. Infectious material is at random displaced from that source cage, and you never reach the whole space. How far you can reach depends on what the diffusion constant is, how quickly the variance increases, and it depends on the mortality rate. So what we did is we did this also for another bacterium, E. coli, which has a much higher survival, like the Strepsuus that Don was involved in the analysis. That also has a much higher survival in the environment. And you see that indeed this one arrives much earlier in time. And so what you really then have is this type of picture. So the blue line is never getting beyond this 75 centimeters, and the red line, so this is the tail of the Gaussian distribution. So the red line arrived there around 4 days, and the blue line. And so if you look at the concentration, at the amount arriving per day at the 75 centimeters, you see the difference between the blue and the red. So the E. coli arrives around 4 days, and the Campylobacter arrives much later. And indeed, we knew beforehand that this 75 centimeters in this kind of experimental conditions was a kind of critical distance. We found out by accident in earlier experiments. And so you get this very big difference just because this is at the edge which you still can reach as this Campylobacter. That's also why not all the animals in the surrounding cages get infected. So we did many repetitions by now from this experiment, and we're quite sure that this is happening and that this is the difference between. So what we're really saying is that this infectious material has been laying around in this stable for quite some time. It arrives there 17 days and later, and it's built up. So it has been traveling quite some time before it arrives there. And one of the consequences is that if you are going to clean the surfaces between those two cages, then you could maybe interfere with this transmission. So this is for us very important in thinking about hygiene and controlling this kind of infections. And we've been discussing with people from the hospital in Utrecht also to make the links to what is happening with intensive care units, where you have a similar kind of problem. The people are there in beds and they don't move. It's not a cage, but they don't move, and they are a small distance from other people that are not yet infected. And so what happens, and also what happens if you're going to clean the surfaces intermediate between these people. So this is the model. So with the same diffusion rate for E. coli and for Campylobacter, and I didn't mention it, with also two different number of animals in the central cage, you get this kind of data fit. So transmission through contamination of the environment is very difficult to model and to control, but therefore it's very important because it's also difficult to regulate. So when we have an outbreak of an infectious disease, we have all kind of standstill things. So we want better to understand what's happening, and also what can we do in terms of hygiene and biosecurity, cleaning, isolation protocols. So for instance, Don showed the table in Sambatimba's paper in PLOS ONE, and he showed that per day this was the rate for the crisis team of per contact. Per contact, this is the probability for the crisis team to infect. So this is a contact, you have to realize this is a contact between a known infected farm, in hindsight of course, a known infected farm and another farm. So we had all these contacts, and then we asked whether the other farm got infected. And like Don was saying, we substantiated that with the genetic data. So this is the crisis time team rate per contact if you go from an infected farm to another farm. And our estimate is that no farms got infected by the crisis time team. I mean less than 0 point something. Whereas one of the other examples, feed delivery, was 4%, and Don pointed to the egg collection at 0.3 per contact. The egg collection really gets into the stable. The feed is some truck coming to the outside of the stable, connecting to a silo outside the building. And the egg collecting people, they come in with trays, bring new trays, and they come into the front part of the stable. But you see this big differences in this context. So this whole idea about hygiene and cleaning and isolation is something that we try to further quantify. So the final conclusions for our reality can be fitted to some of the models we currently have. And in that case, it provides a useful description of what happens. They are not for BSE after the ban, the spatial kernels, but for implementation of new control strategies, and especially for things like biosecurity and hygiene. And so we really need to go to know better what really are those contacts that make that the one unit infects the other unit. And we are looking for ways to find out and genetic information, but also experimental things, because then we can, of course, think beforehand in such an experimental setting that we make it, that it only can be from certain reasons, can help to understand this. So, being 2 minutes past 6 and the drinks are already out there, so I want to thank you all for being here and listening to our presentation. Thank you.
Don't get off that easily. Julia.
In the spirit of looking for mechanistic reasons.
Stand up, Julia.
In the spirit of looking for mechanistic reasons, the work with the diffusion sort of between the cages, what is it that's diffusing and how is it diffusing?
What we think that's really is fecal material that's diffusing. So the fecal material is contaminating the floor. So this are cages on the floor, and so the E. coli and the I did it very quickly because of the time, but the E. coli and the Campylobacter, both bacteria are shed in the feces of these chickens, 10 to the 7 colony-forming units per gram feces, both from the start to the very end. So so both are there in the feces, but of course the Campylobacter is dying down, so it takes longer to reach a sufficient concentration to.
How does it move?
So
Chickens in between, like moving stuff, right?
yeah, so there are people sampling the animals, and and because we can make the comparison because we have the same 2 bacteria in the same animals, but it's of course a function of how people move, how the air is regulated in the stables, things like that. But what we are interested in is the comparison between the two bacteria.
Yes, I just wanted to draw an analogy. So when we're talking about this pathogen reaching out in another individual, so if you think about animals searching for food, if the food is a prey item, so then there is a very well-known relation between how they move each other, one respect to each other, if you want to find it. No? So if there is one which is very fast, you better stay put. You eventually comes to you. On the other hand, if you are fast and then you stay, so you go. So I wonder, if you can think about implementing, within some closed environment, a way to actually make animals move in different way, because the relative movement is very important. Of course, your complication is that you have an object, which is the animal, which has a sort of a veil of, which is the shedding the pathogen. No? But so then you have an object which has a finite size rather than being a point particle, but then you can still think about this.
Yeah, so I didn't mention it, but we really did the model for area sources and for area recipient. We did the whole mathematics in detail for the cage size and the cage size of the recipient cage, and so they're modeling the same diffusion for both. That's basically for the fecal material when mortality is 0, and then taking the mortality into account in the model, then you get the graphs for the other pathogen. But yeah, you can do all kinds of other things. I think, yeah.
So, declaring an interest as having been involved in the FMD and the BSE stuff, I mean, you listed a group of people that should meet, and there's definitely still differences of opinions. I gave a talk on bovine TB about 6 months ago, and the after-dinner speaker was a vet who basically told me I had blood on my hands, both from animals and from vets who'd committed suicide as a result. So it's still hotly debated. But there were additional, I mean, you were already bringing a lot of people together, but you also need the extrinsic factors, which at that time were that the supermarket said they would not sell milk from vaccinated cows. So that was politically taken off the table and said, because basically once Tesco said they weren't selling milk from vaccinated cows, so would Sainsbury's and all the other ones, because that's so sensitive in the consumer environment. That has since been dealt with, I think, in terms of engagement. But also the issue that the EU said that any animals that had been vaccinated would have, at that point at least, would have to be killed afterwards before the UK could regain its FMD-free status.
That's also changed.
Yeah, but at that point it was basically what sort of culling could be done. And from a mechanistic point of view, I wondered, with your transmission matrix, does that vary from FMD strain to strain? Because if it does, presumably you should get governments to pay you to have sheep and cows and pigs on standby, so that when there's a new epidemic, they can test your strain and you can do that calculation for the new one.
Yeah, so like I said, there's lots of heterogeneity, and then the question is, what is the most important one? And that's also, that's more the art of doing experiments. So how do you choose that? And I think that many veterinarians believe that there are big differences between strains, but I'm not convinced. There are no convincing data to substantiate that. So, but this was with the outbreak strain in UK.
Okay, that was the 2001.
Yeah. So most of the experiments I didn't specify, but all the experiments on the diagonal, and assuming random of separable mixing, it's only the diagonal that's important eventually. For the picture, it's important to know also the off-diagonal things, but if you assume that it's separable, then all those things on the diagonal were done with the strain that caused the outbreak in UK. The one that we got in Netherlands and isolated from. That's very close to that.
Unfortunate that it started with a pig farmer who was subsequently prosecuted for his.
Yeah. I've seen the pictures. I talked with the veterinarians that were. I've seen the picture of the pig farm. It was very, 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 very far. Took a very long time to find before that. There were many dying pigs and dead pigs lying around. It was a very bad situation. Not something that you can predict beforehand. I can understand that it happens, and then very unfortunate that sheep in the neighborhood were then transported to a market and went to I don't know how many other places. So that was really making things. It's not to say that somebody is to blame, but you get this discussion. So how do you do it? What do you allow for the veterinarians to use their own expertise, and what do you want to say as a government? So that's also the political part. For the government, it's better. It's easier to say, okay, we'll cull all the susceptible foot-and-mouth disease susceptible animals in a certain radius, then let the veterinarians decide themselves, and then come up. And then it turns out maybe it's not effective, or it is effective. So that's made it more difficult, I think.
Well, the other issue was that there weren't a lot of vets available to be doing additional work, because there were rules that a vet or even a vet student who were being brought in to do surveillance could only be on one farm each day.
Yeah, I know.
And so the biosecurity was such that it was difficult to see where you would have gotten individuals to come on and make these expert judgments on farm.
James, final comment.
Well, I've much enjoyed your presentation. There has been a lot of difference between veterinary epidemiology and epidemiology more generally, but other than the fact that you can do experiments in the host species, do you think this distinction between epidemiology and any other sort of epidemiology actually is entirely false, and we should just be talking about epidemiology?
Yeah, in my book there's only rational things and not irrational things.